Hey, welcome to another episode of the Operation Grow Show, Season 5, Episode 13, this week with our guest Matthew from Debacco University. I hope I'm pronouncing that correctly. Let me quickly just get chat going here. There we go. Yeah, this is going to be a great show. It's going to cover some really interesting topics. And so before we get on to that, let's have a quick catch up with everybody. How's everybody doing? Cascadian, so how you doing, buddy?
I'm doing great, man. Glad to be here. Glad to have Matthew with us. This will be a fun one, dude. Luckily, the spotlight's not on me this week, so I get to ask all the hard questions. Relax a little bit, man. I'm glad to be here.
Yeah. Excellent, excellent. And GMF, how you doing, sir?
Just lucky to be above ground and hanging out with y'all. I'm a fortunate man that I get to do that. So excellent. Thank you for asking.
Good man, good man. Raptor, how are things in your neck of the woods?
Doing pretty well. Very excited for this talk. Got to catch a couple of Matthew's episodes. Specifically, was interested in the PCR videos. I've gotten a couple of questions myself from people who are like, "Oh, how does this work with sex testing?" And I wasn't trained in it formally. I was just trained in how to use a PCR. So it was great, like, to actually have something to point people towards. So yeah, thank you so much for that information, Matthew. Yeah, can't wait to hear what you have to say and teach us.
And so on that note, Matthew, how you doing, sir? Welcome to the show, and we're really glad to have you on.
Yeah, well, thanks for reaching out. It's been great. It's been, we're talking about being all remote here, and it's unique opportunities develop. And Pete reached out, and I said, sure, more information I can get out, the better. More people I can talk to try to get some of the science out here on cannabis. I think all the better. So thanks for having me on.
No, absolute pleasure. I know you've got some topics prepared for the show. Let's see, on that note, yep, I'll just let you go for it. We'll keep an eye on questions that are coming through in chat, and we'll take notes of those. When there's a natural break, or if you at the end of each little piece, whatever, you can let us know, and we can pick up those questions. Otherwise, we'll keep them towards the end, and go through them that way.
So I'm kind of free flowing, whatever you guys want. I know Raptor had mentioned the PCR, so maybe I'll just I'll pull that kind of slide set up, and I'll start with that to kind of get the conversation going, because some people may have seen that PowerPoint or kind of video lecture, shall I say. And again, these are available on Debacco University on YouTube here. So again, welcome to check those out. Appreciate all the subscribers that I do get, and the more word I can get out, the better. My plan is, and again, open for interpretation, but going to go over some videos that already are posted to get some of the topics that kind of was the reason why I'm here, which is very much appreciated. And I'm going to go to definitely end with, or in some way incorporate, rooting success for cannabis clones that I was able to find some scientific papers on, specifically for cannabis, which is always a challenge to find. And that's a video that will be going up, but isn't up yet. So I want to give all of your viewers here a little sneak peek, definitely on that one. Excellent. So let me go through, and I've got sharing screen rights. It looks like I do. Oh, let's go through, and I'll pop that one up. So funny, I work in so many different platforms. It's like, all right, making sure I got the Zoom format down, shall we say. So this one looking at determining sex using PCR. Now, PCR, this might be a little cost prohibitive if you're kind of growing only a couple plants, but however, it is what's done definitely on the large scale. The reason why it's not done on the small scale, simply because of upfront costs. The machines can be expensive, the materials can be expensive, but once purchased and set up, it really does become a very quick and easy way to be able to determine whether you get a male or female plant. And that's what we're talking about here. Cannabis is mostly dioecious, meaning you're looking at producing either a female plant or a male plant. Some fiber hemp varieties have been bred to be monoecious, which means they contain both male and female flowers. But the whole goal is really to kind of, at least if you're not breeding, I should say, is looking at developing those strong female lines and having a lot of those female plants so you can harvest the buds there, of course. So separating those out earlier the develop is useful, because you don't want to be taking care of a plant that's a male plant that you're going to discard anyway. and waste all of your time, labor, water, nutrients, lighting area, everything that goes along with growing a plant if you're not going to intend on keeping that for the long term. So what PCR is, it stands for polymerase chain reaction. So what does that actually, mean? What are these lines? What's going on all over here? Well, it's looking at the DNA, the genetic sequence of the plant. So as a result, the reason why this is advantageous is we can go through and take a seedling, literally just emerged out of the soil. We wait just a little bit longer usually. Take literally a hole punch section of leaf out of the plant, and then we would run the PCR on that. So what the PCR is actually doing, polymerase chain reaction, it's a chain reaction, and it is looking for a specific marker, and it's multiplying that small section of the DNA. Instead of replicating the whole DNA, it's looking, is this little section present? And if so, it's going to go through and multiply that particular section. We're hearing a lot with, sadly with COVID and COVID testing, you want PCR test for your COVID test. Well, that's operating in the exact same format. It's taking that, it's looking for that small section of DNA, like a gene within the whole genome. And if it's find it there, it'll mass multiply that. Why do you want to mass multiply that? Well, because then you can run it, and this is a gel, and this shows you different sizes. So if you get the mass multiplication of that particular size fragment that exists, it'll show up here as kind of a loud line there. And that will tell you that that's present, and that can help you indicate whether you have a male plant or a female plant. So what has to be done is you have to find the male genes. So exact markers can be debated as far as some companies, if you send it out to a company, some use slightly different genetic markers for the male genes, but in general, the concept is the same for all. The markers of the male gene or genes are found in the plant, then it will not produce the high cannabinoids sought in flowers by many growers. It's going to produce the pollen, which might be important for breeders to know, but really for those looking at those cannabinoids or terpenes, not going to be the greatest producer for that. We see the example here of kind of what it would look like. I couldn't find a great cannabis example because a lot of those are kind of under, wraps, shall we say, but it's the same idea here where we're looking at horses, being able to know whether you got a mare, which is an adult female horse, or a colt, a young male horse. What's going to occur is with the different markers, you're going to have these different kind of bands that occur. So you run your sample 1, sample 2, sample 3, and you basically what they're doing, scientists are matching up those markers to see, okay, do I have a male plant or do I have a female plant? And as I said, those markers can be debated, but the concept at least is exactly the same. So again, why use PCR? And this kind of shows a nice little example here. Remember, human DNA. So if you took your DNA, which is kind of amazing, and were to kind of like unravel it because it's kind of wrapped up and stretch it out, and each of your cells would stretch out to be about 6 feet long. So we're not looking at replicating the whole genetic sequence. We're looking at, are those certain genes or markers there? So with PCR, the analysis continues to be more advanced to pick out what's the best gene basically to look for. And as I said, it's a cycle. So you may only need one copy, you really only need one copy, and this cycling will occur and create potentially, if you let it go long enough, it's definitely exponential amplification. From one sequence, you could technically get billions of copies. And that really makes it very easy to detect on that gel that I showed before. Now, you might think billions of copies are going to take such a long time to get to that end result. Really, it's not. It's a very quick process, depending on how many copies you need or theoretically need. You could be looking at doing this in literally a couple of hours from basically taking the sample, setting it all up, running it, and getting your results. So plant sample required is very minimal, which is great because if you're dealing with plants this big, you can easily go through and sample those. And that's great because you don't want to be growing a plant, to, huge size and taking all that time and floor space when you're not going to really utilize it in the end. So if you can determine at this stage, going to cull this one out and keep that one, really makes a grow operation a lot more efficient. So again, the goal, cost and benefit, and this is true for anything. This could be, the PCR testing or the genetic testing or different lighting or different HVAC systems. Can the cost be justified? And a lot of times it is expensive, at least upfront cost, but larger grow operations will often set up this in-house because they can justify that cost over the long term. Because after you get that initial cost, the reoccurring costs are actually quite minimal. So that's an advantage. For medium to small operations, individual samples usually will be sent out to a lab for testing for a fee. Those fees can vary, again, depending what lab you might be using. They might seem a little expensive, but compared to the initial upfront cost, they're definitely greatly reduced from that. So that's why a lot of growers tend to go with the sending it out, at least initially, for those medium to small operations. And then as they get larger, and you can justify the kind of maybe the personnel to be expertise on it, to buy the equipment, to get all the materials, at that point, they could go through and then justify the upfront cost for that. So again, each operation's got to go through their own cost benefit analysis to determine whether that's a good option for them to tell whether they have male or female plants. So that's what I have on there. Let me just stop sharing that for a second and kind of break it up. I don't know if you guys do questions in between or want to do questions in between. It's all good for me, or if not, I can roll right on, but wanted to take a pause.
I know I certainly have some questions.
I got a handful, man. I
know I'm a quick talker, I know, so I want to make sure I integrate pauses, shall we say.
Let the heavy hitters go in. Cascadian, it's up to you.
Yeah, go for Cascade.
So it's hard to find the let's just dive to the bottom of the rabbit hole and come back up for air here. It's hard to find the markers for the male cannabis plants because the people that are doing it aren't sharing that information, then how does one set up the ability to start their own database collecting genetic sequences to figure out where the overlap is? Like, if I was to test 1,000 males and 1,000 females, just random cannabis plants, eventually I'm going to have an overlap in a particular area, and I could say those are male genetics. But how do I set up the system so that I can do that and keep that? You see what I'm saying? Like, how do I build my own? I know it's a big question, but
yeah. So the advantage is that while certain companies tend not to release the markers that they're specifically using, I will have to look. I know I have it somewhere in one of my videos, one of my many ones here, that I did find that they are releasing, now that stateside is becoming, federally, they can do tests on it. They are starting to release some markers, and they're doing comparisons on certain markers, saying that, okay, use marker, and I forget what they're exactly called, but I don't want to quote it and misquote it, shall I say. You're using this marker, well, let's compare it to that marker. And then they determine, okay, this plant is male or female, and then growing them out to term to see how accurate they may be. And I know, and I will have to go through and take a look, and maybe as I'm feeling another question, to find the exact marker that they were able to find that proved most effective. So the information is starting slowly to leak out, mainly because more universities, more areas are doing research on it that is funded by public dollars. So as a result, they kick that information out. Cool. So yes, I will have to find that, but yes, there is at least a good starting point. May not be the quote unquote best marker, but definitely one that can speed up your learning curve, shall we say.
Awesome.
So in terms of the PCR, just a couple of questions, because there's the mini PCR project, which seems to be sort of like an open source project that keeps the cost relatively low. I think that's about $500 for the I forget what it's called, the main part, and all the components. I think it's all in probably for about $1,000 would get you up and running on something like that. And you get 8 channel, and then I think the other one they talk about 16 channels. So is that just it can identify more with the extra number of channels, because of those the lanes, or as they call them, lanes, I think it is.
You're basically able to do more in the same run. So there's usually what in the lab, you're always trying to, what's the bottleneck? What's the bottleneck? Well, there'll be 5 different PCR machines all set up. Now they all do the same thing. And basically it's kind of weird because all PCR really does, the machine, despite their cost, is all they really do is heat things up to a precise temperature and cool things down at a precise temperature at very specific times. What they're doing is simply just causing DNA replication to occur very fast. I mean, it used to be done, and I want to talk a really rudimentary. PCR was developed by someone who took test tubes with different water baths and went 5 minutes here, 2 minutes there, 5 minutes here, 2 minutes here. And now they charge thousands of dollars for machines that do it automatically. So,
So you could use this to establish a basic DNA fingerprint for a plant.
Exactly. Yes. Now, what's really used for is to really identify those certain genes. That's where it's best utilized for.
So this works
Because-
for like sex testing, but does this also work for cannabinoids or secondary metabolites? Or
Once you determine where or what the gene is you're looking for, and you have the primers for it, you are good to go. You can quote unquote find anything, or not find it, or prove whether it's, I should say, prove whether it's there or not.
Gotcha.
And I did find some of the genetic marker testing. So after Raptor goes over his, I did find the other video, and I can share with you with confidence.
Oh, no, please go over. I was just doing like, some people might not be familiar with the tools that's necessary for what we're talking about. And this is actually this is not my presentation whatsoever. I do not take credit for this. This is from, actually, I could probably go up top and give him proper credit.
Damon is there on the front?
Damon Todd.
Okay.
So this is from his mushroom DNA sequencing for the community science. This was brought forward by Humboldt Bay Mycological Society. So yeah, he was gracious enough to let people have this, and just so people can be informed, I merely just brought this up. But yeah,
no, that's
please.
Perfect.
Yeah, and it just goes over that, just a quick relay of all that. So yes, please continue. I just thought of a little visual aid for people.
Perfect. It's that thermocycler, and just as the name implies, it cycles different temperatures. Don't want to take it away. It seemed like it's this great, giant, scary thing. It is a little, like I said, cost prohibitive for some people for kind of quote unquote what it does. But let me go through and let me pull up this other one here now that I found it. Since Cascading was asking, I always hate to cross or mention wrong information. Enough of that occurs, unintentionally. So I have here, this is the genetic testing one, which this one is already actually posted. Let me see if I can swap here. The different parts are having different monitors. There we go. So this is, again, I go over a little bit, again, kind of what PCR is. And like I said, it literally is, well, why would you cycle the temperature? Well, you literally start with that one sample, and that's every time it cycles, you're creating more and more copies. So once you get from 1 to 2, well, now each of those can double, then each of those can double, and each of those can double, and each of those can double. So it's a very, a time efficient process. It's used for other things. It could be male-female genotyping, sequencing, and of course, IP or intellectual property protection. So when we're asking about the DNA markers, this is the actual study that I pulled from. It's from 2016. And they go into more of the PCR analysis in more details. I gave you the proper citation. I give you all the links, all the good information here. This is what we just talked about. Again, determining male or female plants, overview of samples. A lot of people doing different research, a lot of different markers here. Here's some of the actual primers used. And DNA, again, is that T, C, G, and A, those thiamines, cytosines, adenines, and guanines sequences. Those are the nucleotide bases. And it's okay. So what primer sequence? Which ones are we looking for? And it looks like, just a bunch of T, C, A. Well, those are the specific nucleotide sequences there. So looking at male sex-linked DNA, well, this is 119 base pairs, 323 base pairs, 390 base pairs. Those are the number of G's, A's, T's, and C's in the actual sequence. Not all have the same reliability. So this is what they were looking at. They did a bunch of testing, and that thermocycler is what's running through, and then it gets transferred, as Raptor had showed there, on a gel. And this is literally a gel, and it's like a piece of gelatin. Literally, that's maybe that thick, thick of your finger thickness, shall we say. And the little holes here, all the micropipetter, it's where all the DNA is put. And then there's a charge applied, and DNA is a negative charge, so it pulls that DNA through. And you can see how it can be very precise on what it pulls. And this allows for a great and very easy to read comparison. You can clearly see that there's more DNA here than here compared to this sample. So this is what the computers and our scientists are looking at. So what they did is they kind of did this kind of marker testing. And this was their basic conclusions, is that I guess the if it's pronounced SCAR, SCAR 119 marker, appear to be the most reliable for male plant detection in cannabis. Other two showed limited or even controversial results, as we see here, where the green indication here kind of helps show whether or not it got the male or female plant correct. Now, even the markers that didn't work quite as well, you could see that they were reliable definitely most of the time, but at least based on the research published here, that SCAR 119 marker proved the most reliable. And again, they're looking at that genotyping as well, and you're looking at basically finding that one little portion, that one little sequence within the entire genome. And that's where where's the technology really come through? It's really right here. It's in the primer sequence, because that's what you're going to be looking for in binding, to go back to the first couple slides here. So you're going to be looking for in binding to that actual DNA sequence. So as I said, that whole DNA is this long. You're only looking for a small sequence here. If that primer finds a binding site, it'll bind and it'll go through and replicate that PCR process. And then you'll get a reading out on one of the gels here. Now, if it didn't find that binder, you might have nothing in that. You might have everything in one little glob here. So again, it tells you, again, male or female in that regards there. So that gives you just a little bit more of the detail, shall we say. Again, I don't know, again, how detail everyone wants to get into, but that is at least some level there for understanding.
So the SCAR 119, is that something that would be available for purchase then? Like, I'm trying to connect, like, how do you buy your way into the information here?
Of all the primers available, that would be the one, based on that research, I would recommend you go out seeking first.
Gotcha.
For the greatest reliability as far as, what they have. And that's why I always post the full, the link to the article, because you can get, of course, more details in the sense of, if you're going to do this for real, every primer has a slightly different potential temperature that you want it to go up and down to. So in that article there, it'll tell you the exact thermos to cycle, exact temperatures to hit for what duration of time. So yes, that would be the primer. That's where the technology is. It's the primer.
So a primer is a lot like a standard then, if you were doing
exactly.
gas chromatography or something. It's the pure sample you're comparing what you have to. Okay, cool. Strapping my head around all this on the fly, it's fun.
A question from chat. Are you familiar with, I don't know if it's a product or maybe a company called AlphaCat, a kit?
I personally am not.
Okay.
I tend we had someone else was curious about the cost of setup. Like, if someone were to look at one of these kits, just an approximate ballpark, because we've got a number of breeders in our chat that are working on, that medium scale and trying to work up. What kind of investment are they looking at to get a kit available for themselves to start doing this, assuming they don't want to farm it out to someone?
I would say the major cost is in the actual thermocycler. Raptor had showed there, do you have a micropipetter? Well, they can be expensive, but they're not that expensive. A calibrated high-end one, you're talking about $200-ish. So make sure if you are buying one, you do get it calibrated. I can't tell you, I was working in a lab with someone that was kind of coming through doing their research work. And yeah, the micropipetter there, $100, I'd say $100 to $200. I would not go any cheaper, and make sure you spend the money to get it calibrated. Simply because from experience, someone doing research was, and they were doing multiple trials, and they could not get repeated cycles that were showing any sort of consistency. They were getting wild, differences, and they really couldn't figure out why. And they thought it was the gel wasn't running right, or they made sure the temperatures on the cycler were good, the reagents they were using were all new. It turns out, again, and sadly in hindsight, the micropipetter they were using wasn't calibrated. So what does that mean? When they were dispensing X amount, well, experiment one, they were dispensing X amount, and they were maybe dispensing X.2 amount, or X times. It was so variable on what you were adding. And when we're talking about PCR, and I don't have one of the tubes here, but they are, if you're looking at any sort of like size comparison, if you take a pen cap, you're talking that this pen cap is the size of the reagents. The micropipette tube is like the very bottom portion of that pen cap. It is very, very small amounts. So slight differences when you're at this level can make huge impact differences. You know, the PCR tube, again, sticking with the pen example, because hopefully everyone has one to relate. PCR tube is only the size of the very tip here. Now that very small amount. So small changes can make a large change in the end. So you need a good micropipetter.
Yeah, this is a
PCR tubes. They're cheap, 10 cents a sample, if that. The reagents, that's where. Yeah, there we go. Okay,
here's a centrifuge tube. Yeah, and then so the PCR, if you can see, there's a little mark on this thing. The 0.1 is about basically all the bigger a PCR tube is. They're just tiny.
Well,
when I do talks, I always talk about PCR, then I give out the tubes, and they're like, "This is it." Like all that occurs in this little tiny. Cascadian's got it.
It's the tiniest drop, but you would be amazed what that affects. Like everything.
And so from that slide you were showing there, Raptor, it looks like
I'll bring it up again. I just wanted.
I mean, it looks like the thermocyclers run anywhere from 300 to 3,000, which again is going to be, you probably are getting what you pay for, but minimal returns at the 3,000.
You get more lanes. You get some work faster. You don't have to wait as long. It's really on the technology, the newer technology that's more updated. It's going to cost you more, just like a computer. But the very interesting thing about this that Matthew showed in his slides, that the PCR, it could be up to like $3,000, $5,000. But thanks to what I was taught in this demonstration before from these slides, called biohackers. And these biohackers basically took the main PCR design and made it cheaper and use more affordable parts, but kept the basic coding and everything. So just like a computer, just like a computer. So I don't know if any of them are listening, but thank you for making this available to the community, to citizen science. They referred to it as something else, community science. I like all those terms. They all mean the same thing. It just means we come together, and with more of us doing, being educated and doing these kinds of experiments, we can do so much more. It doesn't just have to be on an academic level. We can learn from academia and apply it throughout the community. So that's what I really like what Matthew's doing. He's showing us how to do it at a baseline, and it's the bridge, basically, between academia and the community. So I always think that's a great thing.
It's almost at this is almost the cost where if a number of growers
breeders together
and have the cooperative sort of mentality towards it, then it becomes quite cost effective at that point.
So we're talking what, 200 for the really good pipetter, 5 to 1,000 for the thermocycler. How much are reagents? What are we talking there for cost? Because that's going to be your software. That's the stuff you're using all the time, right? The stuff you're depleting and having to replace.
It's saying that $2 a sample.
It's not.
Depends on the reagent
are the least cost. You just have to make sure they're fresh and you get the right primer.
Yeah, store them. Make sure you don't store them for too long. Otherwise, you're just wasting money. Things like that. Basic lab costs, just like how you would in a kitchen. You don't buy too much of one product, and it's like you buy 2 years worth, and you only need it 6 months.
Vegetarily, you're looking at $1,000. You can get set up at home for this.
Yeah.
Yes. Yep.
Okay. There you go.
A computer.
That answers Magnus'
question.
Yeah.
Just the way lab equipment works in general, all of the cost is usually in setup and maintenance and calibration. So the cost of the test usually isn't that big of a deal, but the hindrance or the thing that keeps people from getting into it is the initial setup cost, calibration, and maintenance. So if you can conquer that part, running the tests on a daily is cheap. That's testing in general. I don't really care what kind of test you're running. It's figuratively cheaper to run the test than it is to set the lab up. So,
and you can come together as a group, as whatever you have it set up and your community set up, and pay lab fees. If you want to be in this group, you have to pay lab fees, and those lab fees goes to paying the reagents, paying the electricity, making sure that if a pipette breaks, you can take that in, or it could be replaced, or that you already have one that's available, so that while one is being replaced or fixed, you can still do your lab work and nothing takes interference. So yeah, just a proper small scale lab setup, like in the corner of your room, let's say like 4x4 tent, keep everything clean. Yeah, it can get you where you need to be. Yeah, and 1,000 is pretty approachable amongst a few people.
Yeah, definitely.
I would say on the Thermocycler, just having used them, if you can get one with a heated lid, it's definitely worth it. Those always seem to work better. I will say it's a stupid little mod. When then close the door, it says a heated lid. The one with the heated lid always got used more because it always worked better. And again, just been my lab setup. But if you're looking at the options, sounds weird, might be worth it.
Definitely want to look out for. No, excellent. Yeah, I mean, my interest is to be able to then, from the breeding side of it, is really to be able to do the identification on a genetic level and be able to say, these are closely related, these are distant, they don't share a large amount of genetic material, or they do share a large amount, so you can make certain decisions. And also being able to when it comes to this thing hitting the commodity market properly, it's going to be, you better make sure what you're selling is what you say it is. And there's only going to be one way of doing that. So you then need to be able to check it against something. I mean, it would be nice to have it at a point where you can go into a dispensary and you buy what they're calling, X, and go, all right, test it straight away. And it'll come back and give you a match and say, it's 99% X, or it's a distant relative. Is that
as
okay?
simple as that is? A lot of that's missing right now, sadly.
And we were talking about marker-assisted breeding. There was one other question earlier from chat: are there any markers for hermaphrodism? Its likelihood, its dom. Because we always have this debate of, is it a mechanism? Is it a genetic trait? Is there any? Has there been, I should say, any kind of work linking those kind of traits or markers?
As far as I know right now, no. I don't think any have been identified specifically for that. Some of it can be environmentally influenced potentially. Again, what needs the genes in there to do it anyway. But I think more of that is a little associated with, they haven't been really identified quite yet, at least in the data that I've seen.
Thank you. Yeah, that was another question we had from the breeder in chat.
Yeah, it would be interesting to compare those monoecious hemp cultivars to our dioecious drug cultivars to see where the genetic overlap is and where the genetic differences are. I think you could elucidate some interesting tidbits out of that.
I don't mean to bring up Satan, but doesn't Phylos already have that info?
I don't know if they have it. I don't know if they have it on the hemp cultivars.
I saw some of their tests, and they do have a hemp footprint, so they can compare. I don't think Phylos is necessarily the devil. I was just making that joke. Sorry about that. Phylos, you're just people.
Oh no, they just shut the bed for everybody else, unfortunately. To be able to do this work, that's the problem. That's what's so frustrating, because you need the community to be sending in, because of the way this has run riot, is you need people to be sending in samples. I mean, geez, just label it, number it. We don't need to know what it is, and then I can actually go back and say, well, it's close. These samples are all really closely linked to each other. Okay, what do you all think it's called?
And they apparently have, for their testing, when you get it back, they have a footprint for CBD. They have a footprint for a hemp variety as well. So you can compare your genetic markers and profile versus those genetic markers and profiles. So I think they have some of that, maybe just not as specific as we're talking, but I think they have that. Yeah.
I will say, just on the hermaphrodite question there, as I looked back at the study, the SCAR 119 marker did show some positive identification for indicating there some hermaphroditic tendencies in some of the lines tested. So that may have some, again, has there been anything identified specifically for that? No, but that might be one that might give you an indication of a plant that might have a tendency to produce the male and female flowers. As I looked back at the information there, they had a couple that did, some hybrids that did kind of show hermaphroditic tendencies, and that marker did seem to indicate that there was a tendency for that. So just again,
Awesome.
at least an initial starting point, if nothing else.
Well, and I think this is the value. I mean, not to, again, advocate for Phylos, and yes, people may not like how they did it, but it comes back to what they're doing, and the information that they have available is very useful. It's if we are going to do what we've talked about, things like marker assisted breeding and actually knowing what we're doing, knowing what we have, they really are the ones who got the, you know, the largest database put together of an actual knowledge right now. So, I mean, hats off to them. Got to respect that.
They dumped a lot of it. I mean, you can download all the raw data, well, a good portion of the raw data, and then it's up to you to put in the grunt work to decipher the raw data. That's
That's the cost.
You know.
Yeah. But it just goes to show a tool is a tool is a tool. You know, it's neither benevolent or evil. It's just a freaking tool, and it's the people that apply that tool that like you can really see the results from. So yeah, well, I don't. Yeah, you're saying.
So somebody in chat saying that they, you know, Flora Nugged in chat saying as a small breeder, the Phylos markers help him tremendously beyond words. So yeah, you know, that's the idea. Is it make if you want to be accurate at what you're doing, you've got. Yeah, this is the best way of doing it. The ground truth is the genetics, the genetic markers, and the DNA fingerprint.
And it's like
my students actually for a class, they go through and look at the Phylos Galaxy, and I always warn them it's going to take a while to load on your computer, right? Depending on your speed of your internet connection, but it's amazing what's there, and a lot don't realize they have access to. You know, I have them compare different strains, and they're like, "Wow, there's a lot of variability," and then they're all calling it the same thing. I go, "That's part of it. I want you to realize that yes, there is some variability there."
Another good resource for that, so we can be, you know, impartial, is EU Seed Finder database as well. That's very good for, and they have a similar one where you can see how closely related they are, what their lineage is, or what the pedigree of the plants are, which is particularly important when you're getting involved in a breeding project, is understanding the pedigree.
Well, and after talking a little bit with some people about Phylos, I kind of came to the conclusion they're kind of like Walmart. You know, you may not like them, you may not want to do business with them, but sometimes it's going to have to be you're going to have to. You know, especially if you want certain. You know, you may not like how they do business, you may not like something about a certain company, but it ultimately comes down to if you want certain things, and that's what they got. They have the largest database. So if you want to benefit from that knowledge, you know, we may not like them, we may moralize about it, but the truth is they have something that the rest of us can really, really use to everyone's benefit. So why not take advantage of the tool that they offer? Knowledge is our weapon.
Yes.
Yeah.
So with that, Pete, do you want me to kind of go into a little bit of rooting success?
Yeah, I think that's particularly useful. And I mean, as we were saying, you know, before we started the show, you're not just taking clones because you're not always taking clones just for the sake of, you know, putting them out to flower. Sometimes it's necessary to just keep things under control. You know, mother plants that get too big, etc., and you need to be successful because you might not be flowering that, and you need to keep it going for another 8 months or so. So being able to do them effectively is important.
Sure, let me go ahead and share this.
Always wait for it to root before you kill the original plant.
No, yes. You don't speak from experience, right?
No, not at all.
Of course not. I know someone that yeah. So this is a video that will come out, will be out, and it goes through. I was glad to find this particular article here. This is the article. I think it was a thesis or a research done, but it was 2018, so still relatively new. And they looked at a lot of the things I think a lot of growers here kind of take for granted or just kind of assume that they know. So just keep in mind there's many variables. We're talking about, you know, rooting and being successful as we can with rooting clones. Growers often wonder what the impact would be of different rooting conditions, but because they've done it one way and they found something that works for them, a lot of times they don't go through and try to have any sort of variables with that. So the goal here, again, like I think all of us, is to kind of like maximize the rooting production. Now, they've looked at a lot of things, and I tried to do the best I could to summarize it here. I'm not going to go into the great details here, but key parts. They were looking at leaf number, cutting position on the plant, leaf tip removal, and the effect of a rooting hormone. I think all kind of 4 main things that a lot of growers wonder about. Well, how many leaves do I need? Where should I cut them? What if I cut the tips of the leaves? What about rooting hormone? What should I add for that? So that's what they looked at. They also were very good at keeping their conditions consistent, which is another kind of potential variable for a lot of growers. Do you know what your relative humidity is? Are you keeping track of that? What is your temperature? What is your light intensity? All things just any grower looking at propagation should be keeping in mind. And of course, this being a scientific study, which is what I love about that, is they quote their materials and methods. I usually read the abstract, and if I like the abstract, which is like, think of that like the quick summary, most people go from the abstract directly to their conclusion or discussion. You want to go from their abstract, which is their summary, to their materials and methods. And the reason why you want to do that is just for the points mentioned here. Did they have a good idea of their relative humidity? Were they tracking their light intensity? Were they doing all the aspects of a typical experiment correct? Because if I read that and I feel that they did not do a good job with their materials and methods and process, their conclusion becomes not valid to me. So that's why I read the materials and methods. Okay, that looks good. Now let's go and see what they actually concluded. And they came up with some great, some great comparisons here. And they were looking at the biometrics. This is not in a picture from their particular study. I just pulled some images here. They were harvesting their cuttings 7 days after, 12 days after, looking for visible roots here. And what they did is this is actually from the study. They were kind of making an assessment of how well a successful rooted cutting or clone would be. They gave it a root quality index, an RQI. They basically said you're a 1 or you're a 2. 1 is, you could see here, rooted, but not really great. 2 is kind of what you're looking for, the top tier there. Before RQI measurements, the substrate was washed in rooted cuttings with RO water so that they were getting a very consistent visual assessment here of the particular clones that they were going to be looking at and investigating. So this kind of gets into the great part. This is like the data here, and they're looking at, again, leaf number, position, leaf tips, and rooting hormone. Now, when we're looking at bar graphs, just to kind of give everyone kind of the same standpoint of when you're looking at a scientific bar graph, in this example of leaf number, they're looking at basically 2 fully expanded leaves, I believe, or 3. When you see, 3 is higher than 2, well, when you see these letters, basically if there's an A and an A, this shows the degree of variability, because as you know, they were looking here at 80 different clones. So there's going to be different variable of success. You expect some variability, that's okay. Variability is a good thing because they're plants. They all don't behave exactly the same. This one does look higher, but compared when you look at the statistics, based on the degree of variability that these had, these would be considered to be statistically at least the same. They're not different. So even though people will always look at the bar graph and go, oh, that one's taller, that one's better, statistically, they're both within the same degree of variability, the same range. So the leaf number, 2 or 3, their study basically showed didn't really make a difference. Basal cutting or apical cutting, we take those cuttings at the very top of the plant. Those are our best areas. Those are the best clones. They're the best success versus the ones on the side. Well, by their study, didn't really make a difference. So it doesn't matter if it's that top clone or that side clone. It's going to be have the greatest chance at, or percentage, I should say, at rooting success. This one is interesting, the leaf tips. Cutting the leaf tips or not cutting the leaf tips. A lot of growers I know always go through and they cut their leaf tips. They want to reduce the transpiration on the plant. Well, what they don't realize, I think, that they're doing is they're stressing the plant by cutting the leaf tips. They're reducing the area. They're causing water to be kind of leave. They're causing callus to try to form so the plant can seal itself. And what that ultimately translates to, sadly, is a reduced rooting success. Now, I know a lot of growers are going to argue with me and say, oh, I cut the leaf tips and I get 100% all the time. Well, are you doing 80? Are you doing enough? Are you doing large scale? This says to me that really, this is statistically different. Don't cut your leaf tips. You're going to get a greater rooting success rate if you don't cut your leaf tips. And looking at rooting hormone, organic versus synthetic, I know some people are under maybe organic restrictions, but definitely the synthetic IBA, which is an auxin hormone, did make a large difference, better at improving rooting success here. And again, they looked at some other things here. Again, looking at some more cuttings, look at some more chi-square analysis, some more details. But again, the same thing comes here, the root quality. So sorry, the first one was looking at the rooting success, the percentage. Now we're looking at the root quality. And we could see here, leaf number again, and synthetic or organic rooting compounds still held true and made a difference. They look a little bit more even visually, but the statistics also come out all the same, except for, in this case, leaf number. And leaf number, leaving it 3, was slightly better for our root quality. So keep that in mind. Personally, I like 3 leaves and not cutting the root tips and using a synthetic rooting hormone for the quality difference. And again, they did more comparisons here, cutting or uncutting, diving into that a little bit more deeper here. And if you do cut the leaves off, the apical clones, which are the ones more towards the top, the apical merisms, the very top ones, did have a slight improvement. But if you're looking at the basal or apical, statistically uncut, there's no difference there. Here they only looked at a sample of 40, but that's still a pretty good number to develop some worthy statistics from. So what's my general summary here? Rooting hormone, 0.2% IBA gel. Again, Clonex, there's different ones out there. I just showed an example here of Clonex. Derived higher rooting success rate than a willow extract, which is the organic one. Cutting 30% of the leaf tips reduced the rooting success rate. Number of leaves, 3 or higher root quality compared to 2 leaves influenced the rooting success. And the position on the stock plant did not really influence either rooting success or the root quality overall. So again, that would be worthy to go through and do. So as I do try with all my videos, I try to go through and give you that maximum success. What's the take-home message? Take your plant material from any part of the plant as long as it's healthy and in good shape. Make sure the cuttings have at least 3 fully expanded leaves. Do not cut the leaves and add IBA rooting hormone. That's my quick general summary. And I know I do pull pictures from a lot of different growers. The one picture actually here is a student actually in my office, which is always a unique kind of aspect to have people walk by your office and have actually growing cannabis plants. But the students do go through a rooting experiment as part of the class. They learn about it, and they're actually able to go down to my office and actually complete that, which they seem to always to like, and kind of prove to them that you can do clones and cuttings, and a lot of them it's the first time dealing with a plant in general. So always great to share the scientific information, but then to get to actually show some of the students I have exactly that they can go through and do it themselves.
And that point about not cutting the leaf tips in a commercial setting, that's a massive time saver. And don't underestimate, somebody not having to now cut all the leaves on all these plants. It at scale,
it made it better. Great, difference. You could justify the labor, but it doesn't. So yeah, I'm a very much about efficiency, that if it is better, it takes a little more time, definitely I'll go through and do it. But in that case, I think too, if you're looking at selling your clones or, dispensing to people, I think they look better too without the leaf tips cut, and you're getting rooting success. So to me, it's just a win-win. But you're like, well, why don't you cut the leaves? I go, because it makes it root worse. And like, well, how do you know that? Well, I've got data to prove that here.
Plus, there's the possibility of the contamination issues. The introduction of pathogens, you're opening up the tissue. Mm-hmm. So unless you're completely sterile all the way, you know exactly what you're doing, you're just adding more variables for you to go wrong.
Mm-hmm.
Oh, Fruity, one day too late for the information. Hey, buddy.
Yeah, really.
Next time.
If I was to get nitpicky about that study, there's only one thing I would have changed that would have increased the data that you would pull from it. It's kind of sad that they only tried hard enough to do the 0.2% willow extract when what they should have exist. Well, what they should have did was they should have measured the amount of hormone in the willow extract and matched that to the amount of hormone in the synthetic preparation, because that's what you're measuring is the hormones, not the percentage of stuff. There's a lot more in a willow extract than just the rooting hormone. And you're ending up with a fraction of the willow rooting hormone to this 0.2% IBA. And that's the only part that doesn't seem like a fair comparison, which seems like it extrapolated the results.
I'd have to go back and look at the actual, data they said. I believe when they did a comparison, they both said that they had 0.2% of IBA or of their rooting hormone, but you are right. It does not take into account the other components within the willow extract.
That's my only like nitpick about other than that, the information there is beautiful. I wish I would have found the study myself, but I found it now. So thanks.
Yeah. Hey.
But that just from a scientific lens, that's the only thing I can see in that whole thing that would be a question mark. I just wanted
for those looking for that actual study, because I know I went through it quickly, I believe tomorrow, Sunday, I should make around 4:20 New York City time, Central Eastern time zone. That video will be going live on my YouTube channel, so you can by all means wait a little bit, but it will be publicly available, and you can go through and look at that study and nitpick it and see what they did and glean some information from it and find more. That's the whole down the rabbit hole, I always call it, where you find one study and you look at their resources, you're like, oh, they pulled from here, and let me go look at that one and that one and this one. And you got to enjoy the process.
Just compound.
Exactly.
Yep.
That's how my computer filled up with white papers. That right there.
Yep.
We did have one more question from chat on this. And I noticed too, looking in your office, there's no dome on there, right? So we haven't, so there's no addressing of even using that in these studies, right?
Correct. Yeah, on mine, that's one that I showed there for the students to make it easy. It's in a very small grow tent, and it's easy cloner is what I'm using there.
Gotcha.
Yeah, that picture isn't from.
So do you have any opinion on domes? Are they?
I'm sure they were using either domes, or I believe they were using growth chambers, where they were dialing in the specific humidity levels. I just threw a picture there to kind of show the rooting that I do in my class, so to speak, but that was not directly from the study plans.
It kind of depends on how you're doing it. If you're doing it in a sort of a potting soil medium, they're going to need a dome. They're going to need high humidity. But if you're doing it in like an aeroponic or water bath type situation, they don't necessarily need the clean humidity dome. So it just kind of depends. The picture he shared there was an aeroponic setup, and if you're doing it in soil, which they mentioned washing the substrate away with RO water, so they're probably doing it in some sort of a substrate that's going to need a dome. And then they washed it away and looked at the roots.
And just a word of warning as just a word of warning as a manufacturer of environment monitoring devices, do not put your hygrometer in that chamber to check if it's at 95% humidity. Do not do that. You can do it once, take it out straight away. Do not leave it in there, because it'll saturate, and then you have to buy another one. They're not really designed for being left in high humidity environments. If you've got a dome on it, assume if it's got condensation on the dome, you've got very high humidity.
Looking back at the study, Christian, they were using Promix PG organic growing substrate.
Soil is peat moss based core. Yeah. Cool. The other thing that would have been cool to see them do, sometimes I'll have a plant grow an incredible amount. I'll need to trim it and take clones at the same time. So I'll trim it, look at the piece, and then turn that clone or that piece into 3 clones or 2 clones. So it would have been interesting to see them take a clone that had the central stem cut for the root, but also cut because there was more that was above it, and see how that affected the sort of straw mentality with cutting the leaves and losing the siphon and having to build the callus, how that would have translated into all of that would have been really interesting. It feels like they could be another experiment on more manipulations that we play with.
Mm-hmm. I agree. And that's what I love about the science, is it never ends. So you'll hate about the science is you do this, and what about this, and then what about that, and then 10 years later you're like, wow, what happened?
No, it is. Yeah, no, it's very interesting.
And I think that's, that's part of it, is just you got to start somewhere. And, a lot of studies been done in other plants, analogs of like tomatoes, for example. That's great for them. But what's specific about cannabis? It is its own plant. It is unique, and some things are translatable. So that's a good starting point. But now let's try to develop some more research, some more sharing of information specifically on cannabis, so we can make, develop what is the quote best system. Just because you use it and have success doesn't mean it's the best or the most efficient. Share some information, and that's really what it comes down to, and trying to eliminate loss, because especially when you're taking clones, cuttings, like I said, it might be your one plant that you really need to carry those genes over. And you may not be, getting into flower at that particular time, but you simply want more of it. Give it to your friends. You want to be able to, have it not growing in just one growth in one spot,
Yeah.
Keep those genetics going in the gene pool of things. So.
And have you, in terms of taking clones and cuttings, etc., when it comes to tissue samples and using tissue culture, I should say, as a means of preservation as well. I know it's not something we're necessarily sort of scheduled to talk about.
Specifically to tissue culture, I think that's definitely being done a lot more. It's definitely the way to go. It definitely allows you to take very small samples and extrapolate plants from. I will be upfront and honest that I have not particularly gone down the tissue culture route. That's very sterile conditions. It's very lab oriented, very cooking book style, figuring out hormone concentrations. I deal a little bit more, I call it, with the macroscopic scale of the plant. But I know researchers that are doing tissue culture, and it definitely, one, it's amazing to see them work just in general, and not just cannabis, but tissue culture in general, how quick they are in all the hoods, and they can undo the caps with one hand. And I get there eventually, but just to see them have 3 arms, it seems like. They're definitely very good at what they do. And there's a lot of research, I think, to be coming out under that topic in particular, because it is definitely opens a lot more opportunities up in general.
Yeah.
Yeah.
I mean, I would be curious if it's one way to clone an autoflower is to go via tissue culture, because it's kind of the great reset, which is the thing about taking a cutting. It's not a clone, it's just a cutting. It's an extension of that original plant's age and everything that goes with it. But when you get down into tissue culture, you break it all back down into primordial soup and let it rebuild. So you're sort of resetting the clock at that point. Just as a thought, is it a possible means to do that? Because then it could be, that could then make autoflowers a bit more interesting for some people in terms of, oh, okay, I can keep this going now.
Right, you would then end up with a phenotype you could actually propagate or trade, or maybe because you're playing such a hormone game with tissue culture. You might be able to. That's interesting, Pete. Jesus, I didn't need this today.
Can you get the shoots and roots to form without messing up the terpene profiles and the cannabinoid concentrate? Yeah.
I guess that's going to need to be done.
Yeah.
Well, good experiment then. Good experiment.
It's the fun part of science. You come up with an idea, you're like, wow, wait, that initially sounded crazy, but when I heard it again, that might be possible.
That's like 90% of my life.
Yep.
Can I eat that? Maybe. Well, wait, can I eat it? Let's think about this. Well, I can tell you what, I got a dumb question then maybe you can weigh in on, because I learned a technique for sexing that I use, and it's very rudimentary. All I do is actually start them off in 12-12 light, and then I don't actually, for the first couple of weeks when they're sprouting, and then I switch them to a full 18-6, and I can usually see sex traits very early. I know that has nothing to do with genetic testing, but that's just a really cheap way that I do it at home, just so I know what I'm messing with at any particular time.
Sure. I have people that go through, and I call it the quick clone and flip. You get a little side branch, you cut that, you put it in your cloner, which is under 12-12, and you just keep track, keep your records, know who's who, and put it under 12-12. And as soon as you see those flowers, you go back and you cull out that original plant or not. So,
see, and I just don't do the clone. I just do it to the original seedling. I just put the actual
early
seedling
on
in 12-12. Yeah. And then under those first 14 to 30 days, depending on if it's a super long flowering sativa, I can go up to 4 weeks and 12/12 when it sprouts. But if it's something that's, you know, used to a short cycle, I'll do 2 to 3 weeks tops, and then flip it back to 18/6, and it'll have sex traits showing the entire time it's in veg. It'll have 1 or 2 little pistils, or it'll have 1 or 2 little immature balls the entire time it's doing its, you know, vegetative growth.
I mean, morphology, it seems like it would work. As long as you're not cutting your yield down on the end or something in the plant at all, that can work.
I figure worst case scenario, it's maybe stunted by a week, but you know, that's a worst case. But
at least you knew what plants are male. Get rid of them, saves you labor in the end, right?
That's what I'm thinking. But thank you for weighing in on that. I appreciate it.
No problem.
I'm a cheap DIY kind of guy, so.
Yep. I border on the, you know, the access to the $10,000 PCR thermocycler, you know, and then the person looking at, you know, growing a couple plants at home, trying to figure out what to kind of relay to. It's easy to spend money.
Well, the first thing when you say thermocyclers, okay, how can I get my sous vide machine to fucking do that same job?
Listen, PCR was the poor guy doing the water baths, going from one to another to another to another.
Right?
Yeah.
Well, just back on methods, like the earlier methods of all this, and you'll see they're doing a lot of stuff that we have machines for, just like Matthew was saying, manually. That's what cost thousands. You had to have someone back and forth. That was just their job, to just go through on all the lanes. And you're not far off, GMF. Just like with tissue culture, yeah, you can. We can do all of it synthetically, but it didn't start synthetically. It started organically, just through examining and looking through the compounds of, let's say, coconut water. You know, that can be used, and aloe vera, all those things, willow bark. That's why they use those things in the experiments. Yeah, it can be approachable at any scale. Yeah, thank you so much, Matthew.
No
just a wealth of information. I learned so much, and I didn't expect to get the first question in, and it just took up 30 minutes. So it went into this whole wormhole, which I loved. I loved every single second of it, just because it's something I've been wanting to talk about, but there's only so much information already out, and so many people even looking into that info. And then how many of those people are actually involved in cannabis? So there's a very, very small, like, pinhole opportunity of discussion. So yeah, thank you so much for that.
Oh, glad I could provide it. And like I said, Debacco University is a YouTube channel. Welcome to check that out. Always, you know, encourage people, put comments in. I try to keep up with the comments as best as I can. If you've got questions there, trying to get new videos out. I've been doing a new video every day for the last, I think, going on 3 weeks now. Trying to keep the content going. It's getting harder and harder. There will be plenty of content, but the daily updates and uploads is takes a lot of time. As I do try to read the articles and develop a summary. Again, I give everyone, for where I can, the articles, but it does take a lot of time to kind of break those down. And don't worry, they're all not scientific articles. I do try to balance the PCR end, the scientific end, and then I call it the clone and flip end. Understanding I've got a plethora of growers. I do something on mechanically planting your cannabis on a field, you know, which is, I know, a limited market set. But then I also go through and how to germinate cannabis seeds, if you want just some of the basic information. So I do try to border everything. I also got biology and forensic science. If you get recommended postmortem changes in something, like, it was people comment like, what's this video recommended? I do forensic science and I do biology as well. So it's all run through the same channel.
Dead bodies make great fertilizer.
I know, yeah. Ashes to ashes.
Organic.
Yeah, it's organic. Yeah.
Wouldn't it be like
Or
ashes to
real vegan.
ashes, soil to soil, or something like that?
Yeah. I don't want to keep it to around an hour, which I know it's amazing, but it's like it's gone by already. So again, I can talk, I can keep going, but want to be mindful of everyone's time. And thank you for having me, at least initially here on the live stream here.
Yeah, no, it's been absolutely great having you. And what we now usually do, I mean, we do usually keep the shows to an hour long. Is generally how we like to do it. But we do have an after show Hangout now, where it's no longer live, and we allow people who were in chat. I'll post up the link, and they can jump in, and they tend to hang out and chat. And you're more than welcome to stick around if you want to. Or I know it's till Saturday afternoon. If you've got things you need to be going doing, that's absolutely fine as well. But if you have the time,
stick around for a little longer. That'd be perfectly
great.
fine.
So yeah, I will actually just drop that link in.
Once in a while, we get some people from chat. They don't necessarily want to come on and show their face or anything like that, so they'll hop into the after chat, and so it's a really nice experience. Honestly, I really like when people come by and visit. We get a great chat, I have to admit. We're very lucky.
Yeah, so I tell you what, on that note, where can everybody find us? Truby, where can everybody find you, sir?
Yeah, so the best one right now, literally through my YouTube channel. I know I don't have a lot of other social media accounts right now because I'm really trying to divert a lot of time into my YouTube channel, Debacco University. So check that out. You can reach out to me there. I'll answer questions. That is probably the best way. And that's basically how Pete found me to bring me on the show here through the YouTube channel. And like I said, I got a couple more videos going out, definitely in the short term. So that's going to be good. And I got future, just a little teasers. I got stuff on lighting and CO2 for the indoor growers. I'm also going to be doing kind of some soil test reading. I'm going next week to an actual soil lab and going to videotape or record kind of for those outdoor growers that send their soil to a lab and get a piece of paper back. I'm going to actually show you how it goes through the soil lab. So I'm going to be preparing some of that information as well. So kind of boring the indoor and the outdoor growers because it's all plants, all growing. The whole goal is to provide you with information centered around cannabis where I can, and hopefully help everyone out with a successful grow.
Excellent, excellent. And Tyler, where can we find you, buddy?
Find me on Instagram at Shooby-Doo. You can find me on Cannabis, I'll be growing buds, same Facebook.
And if only you'd known about not having to cut the leaf tips off.
At least an hour worth of time, so at least an hour. Gilmore did night, so I'll save some time.
Yeah, exactly. And I mean, I think that's a big time saver. It's, don't underestimate these little things. Time is money at the end of the day. Cascadian, where can we find you, sir?
Cascadian Grown on Instagram. If you really want to try to keep up with me, you can find me at the Heirloom Approach as well. That's my new endeavor here. So, yeah, man, you'll find me here next Saturday.
Excellent, excellent. And TMF?
You can find me on just IG if you want to holler at me. If there's anything I can do to be helpful, Genetic Memory Farms. You can find me hanging out here with Pete on Operation Grow. And, I'm going to try to make fumis tonight. My sleep schedule's thrown off enough that I think I'll be awake all night. So I got a couple of new Kool-Aid kombuchas to drink, so those will be fun. That's about it. So, just hanging out with all these good people in chat, man. I think I'd like to say a shout out to chat. Thanks for coming by. I think we're very fortunate for the caliber of people we get to hang out here. And people tend to, I think, we get some good questions, people chatting back and forth actually on this topic. So, I'm always very grateful for everybody that comes to hang out. So thank you, chat. I really appreciate it. And if I can be of service to anybody, holler at me on IG, Genetic Memory Farms. I'm always baking, fermenting, cooking, you name it. So holler at me.
Excellent. And Raptor?
Yeah, you can find me at Raptor Grow on Instagram. Same way it's spelled here, all lowercase, all one word. And yeah, you can find me on this show at Operation Grow. You can also find them on Instagram. And if you aren't already aware, it's on YouTube. So yeah, it's 1 p.m. Pacific Standard Time, West Coast for me. We're actually starting up a new show called The Neighborhood Grows, and it's going to be shown up on 420. And here's one of the panelists, Mr. Bagseed. Hi, how's it going, Mr. Bagseed? Yeah. So again, it'll be on 420. Please come join us all. It'll be at 4 o'clock Pacific Standard Time. So hope to see you all. It's going to be a lot of fun, and we'll have so much more. Oh, and every week afterwards, it'll be at the same time, but on Mondays. So yeah, it'll be fun.
Cool.
Hope to see you, everyone.
Excellent. And you can find me at PL Grow Systems on Instagram, as well as at Operation Grow 2019. Yep. I know. And here you'll find us here every week. So join us again next week. Once again, thank you very much to Matthew for joining us on the show. That was really informative.
Glad to be here. Thanks for having me.
Certainly generated a lot of questions.
Hopefully a couple answers though.
yeah, absolutely. And if you'd like to jump into the after show, anybody in chat, the link is there. You're welcome to join us. And yeah, so on that note, hope everybody stays well.